Okay, hello everyone. I think that my presentation will give you a little bit different perspective. This is because it will be devoted mainly to the drugs. This is because I'm a pharmacist by training, not a toxicologist, nor I have any experience in the chemicals. So, and the maybe more important reason for focusing on drugs is because the current applications in the regulatory context of PBPK models are mainly in the pharmaceutical industry, and this is the place where the PBPK models are most advanced so far. So let me start with saying something which may be not well known fact, that is the PBPK, which states for the physiologically-based pharmacokinetics, is nothing new. This concept is suggested that its origins of this concept go back to the work of Theorel as early as 1937. And the first paper, which has been published with the PBPK in its title, appeared in 1977. So it's almost half of a century of PBPK modeling history. But very first applications of PBPK modeling deal with the issues related with anesthesia and with risk assessment of environmental toxicants or pollutants. But if you look at the results of the study by Bogesi or Tan, you may notice that over the last 2 decades, the number of papers considering the PBPK modeling used for non-pharmaceuticals has been almost at the steady state, while at the same time the papers considering the PBPK modeling used for the pharmaceuticals rise almost exponentially. This is mainly because the increasing cost of the drug development, also the availability of new data and new methods for validation of the model, and also the availability of the commercial software platforms allowing model building for many people. Also, the global regulators like FDA, like European Medicine Agency, like Japan's Pharmaceuticals and Medicinal Devices Agency encourage the use of in silico modeling and including the PBPK modeling. So, so what PBPK model is, very, very quickly, because you already know that. So the PBPK model is a mathematical model consisting of tissue and organ compartments, which are connected by circulating arterial and venous blood systems. The PBPK model integrates the information on drug with information on the system within the context of the clinical trial design. And in order to provide the prediction of the concentration of the drug over time in tissue and in plasma. And this prediction take into account the rate of absorption of a drug into the tissue and plasma, its distribution, its elimination, and its metabolism. And the fact that all these processes are result of the interplay between the physiochemical, physiological, and biochemical parameters which are connected to the drug and the system. So the information gained through the PBPK models is primarily aimed in providing the detailed insight into the pharmacokinetics behavior of a drug. But their applications go well beyond just predicting PK properties. The PK models offer the possibility to investigate many intrinsic and extrinsic factors and their influence on the drug exposure and drug response. They are also used to optimize, to plan the clinical trials. They are used to select the optimal dosing regimens, which you could do yourself yesterday during the workshop. And they also allow the extrapolation across different populations who are particularly challenging or maybe not feasible at all at the clinical trials, like the pediatrics, like pregnant women or organ-impaired patients. The regulatory applications of the PBPK models have been investigated by at least two authors. This is Dejameh and Lizan, and they find that the most prominent area of application in the regulatory context of PBPK models is the prediction of effects of drug-drug interactions, and this is followed by pediatrics and also the hepatic impairment patients. This is mainly because these are also the main application areas in the industry, and the reason for that is also that there is a regulatory guidance on PBPK modeling in these particular areas. So a few examples of regulatory applications of PBPK models. The first one is the drug-drug interactions. In the November 2013, so quite recently, we could witness the very first case where the PBPK modeling was used in lieu of conducting a clinical trial to make a statement in a label of a drug. So, so far the PBPK models were used to decide whether to conduct a clinical study or not, to answer different regulatory questions. But this is the very first case where the sponsor decides to put something in the label of a drug, which was supported only by the simulations they conducted, not on the clinical trial. And we have many examples that follow this Imbruvica case, where the PBPK models were used in lieu of conducting clinical trials or were basis for request waivers for clinical trials, especially for the drug-drug interactions. And one of those examples is Eliglustat, where the sponsor decides to use PBPK modeling to support the dosing recommendations for patients being poor, extensive, and intermediate metabolizers, as well as for combinations of this drug with the CYP2D6 and CYP3 for strong or moderate inhibitors. And as a result of PBPK application, only 2 clinical studies were sufficient to support labeling for a drug, and all the remaining 12 scenarios for possible DDI interactions were addressed only with the simulations. The use of PBPK models in predicting and assessing the consequences of drug-drug interactions were also recently strengthened by the new draft guidelines for the industry issued by the FDA, where the agency for the first time deems the modeling and simulation using the PBPK model as a type of a study which can be used in lieu of some prospective DDI studies. So a huge progress in the acceptance of PBPK modeling. Another application area for the PBPK models in the regulatory is in the special populations. In this area, the PBPK models offer the viable alternative for clinical studies. They offer a possibility to test and simulate and test the scenarios which could be never tested clinically. So the current guidelines by both FDA and EMA primarily focus on the healthy volunteer populations, with the EMA guideline providing some additional guidance on modeling in pediatrics. But in general, regulatory requirements or guidelines are lacking for other special populations. But this does not mean that the special populations and models for them are not constructed and used in a regulatory context. If you look at the results of survey done by Yoshida on the appearance of PBPK models in new drug applications at FDA, and you compare the proportion of labels of the drug with the PBPK in a label for all drug categories with the category of anti-cancer drugs, you may notice that there is a somehow wider acceptance of the modeling in this special population. And there are several reasons that could explain that. These are the problems with the recruitment of the patients. These are the ethical or safety issues in this special population. Also, the narrow indices for most of the anti-cancer drugs with the high toxicity, which may impose the need for precision dosing for these patients. Also, the fact that the oncology patients usually take many drugs concomitantly to treat the comorbidities, to treat the side effects they are having, and this already fragile population is faced with the increased risk of drug-drug interaction. So probably this is the reason why the DDIs are again the most prominent area of application also in the oncology patients. But there are also other issues in this special population which can be addressed with the PBPK modeling. And one of them is the pediatric oncology, where the PBPK studies demonstrated their ability to sufficiently and properly predict the pharmacokinetics of the drugs in children, and their ability to support clinical trials, enable first in pediatric trials, and select the proper starting dose for children. They also demonstrate the possibility to predict the drug-drug interactions in the children. So, as you could see, those two applications, so extrapolation to special population and DDIs, are the most prominent area of regulatory applications in new drug development process. But PBPK models are also applied in a generic drug development process. And the example is the bioequivalence study, where the PBPK models may offer many advantages over traditional studies. The example of such a study which demonstrated the capability of PBPK models in bioequivalence study was done by DOCI, where the formulation-dependent effect was and its influence of on the bioequivalence study was analyzed. The authors compared the two formulations of for levothyroxine with the different pH sensitivity for in vitro dissolution. And they used the model to predict the bioequivalence outcome. And with the use of PBPK model, they demonstrated that this approach is able to predict properly the bioequivalence outcome in healthy volunteers, but also the results of the study suggested that in some special conditions, which in this case is achlorhydria, may change the exposure at the target population. And that's why the conclusions drawn on bioequivalence in the healthy volunteers and target population may be wrong. So this study suggested that the patients taking the proton pump inhibitors or Japanese elderly, which have higher frequency of achlorhydria, may have changed exposure for a drug outside the bioequivalence limits. So this study shows that the proper implementation of PBPK modeling can postulate in vivo consequences of any difference between the formulation, the tested one, and the reference formulation. And it can do it not only in healthy volunteers, but also in other types, let's say, populations which are not commonly assessed during the bioequivalence studies. So these are main examples of the PBPK applications in the regulatory area, but we can expect that it will be further expanded, because if you look at the results of analysis of PBPK modeling applications in the industry and in the academia based on literature data, you can see that the DDIs cover only 28%. So this is somehow different from that what we've seen on FDA submissions. So we can expect that all those new applications also appear in regulatory submissions, and the areas will be further expanded. So together with the increased use of PBPK models in the industry, also the regulatory bodies have been faced with the increased PBPK-based submissions in covering many intended uses or application areas. And what they observed was the significant variability in the format and the context of the PBPK reports. Both the agencies, the EMA and the FDA, were concerned with the fact how the PBPK models are qualified and verified, and they are also finding that the model qualification for the intended use is poorly developed, and that the PBPK simulation reports are deficient in details. So they both decide to issue guidelines on PBPK modeling in order to somehow standardize the process of reporting of PBPK analysis results. Also, the pharmaceutical industry decide to form a working group with the aim to develop the best practices in PBPK modeling development, and also their application and their documentation to provide more consistency in the reporting and conducting the PBPK analysis. In addition to the format of the reports, additional emphasis, especially in the EMA guideline, has been devoted to the qualification of the model. And both the qualification and verification are the indicators of how confident can we be with the model predictions, because we want to know whether there is a sufficient scientific support for the certain platform and for the certain application of the model. And the documentation of the qualification and verification process needs to cover all the three components of the platform, which is the computational framework, which is the physiological framework, this means the system, and the drug model. And this means that the user or the manufacturer of the tool needs to ensure that the particular platform and the relevant databases are suitable for this particular intended use of the platform. Also, the documentation of the physiological framework needs to include all the equations used to describe the system, as well as parameters used to fit the model. And the drug model verification needs to document the compliance between the input parameters and underlying mechanisms, and also the ability of the drug model to recover the clinical observation needs to be provided. In addition to qualification and verification, also sensitivity analysis is required, especially for the parameters which have high uncertainty or have high influence on the PBPK modeling results. And the extent of this qualification and verification of the model is related to the impact that model and simulation and its result have on the regulatory decision. And of course, the higher the impact, the greater the expectations and requirements for the qualification. And the regulatory impact in case of drugs is directly related to the risk to the patient in case the modeling and simulation or assumptions for the model lead to erroneous regulatory decision. So the decision whether to accept PBPK analysis result instead of clinical trials and clinical data is made on a case-by-case basis. Making these decisions, we consider the intended use of the model as well as quality of the model, its relevance and reliability of the results provided by the PBPK analysis. As I mentioned at the beginning, the applications of PBPK and PBPK concept stems mainly from non-pharmaceutical areas, but the majority of the work in developing and the verification of the approach occurred in the pharmaceutical industry. There are other sectors apart from the pharma industry where the PBPK models are already used, but they are, as you can see, they are rarely used for the regulatory purposes. And it's all about the mentioned by Matthias, the confidence in the model's predictions. This is the same case as with the drugs. When we model a risk for a chemical, the implementation and the results of the analysis of PK modeling may have high influence on the decisions and on the population. So that's why the models need to be properly verified. They need to be verified for their structure, for their biological characterization, for computational implementation, and for the predictive capabilities. And best practices for model assessment have been proposed some time ago by World Health Organization, but I'm not aware of any regulations or guidelines specific for the PBPK use for chemical, for assessment of chemicals, apart from a white paper from Environmental Protection Agency considering PBPK use in the assessment of acute exposure. So, sorry. So there is lack of guidance coming from the regulatories, and there are also some other issues that have been raised as barriers to the wider acceptance of PBPK modeling in health risk, public health risk assessment. And the major one is this already mentioned confidence in model prediction. The inherent requirement for the validation or verification of the model is the need for data, for clinical data to verify the model. And PBPK modeling in the pharma industry benefits from the fact that clinical data on drugs are generally available, which is not the case, or which is rarely the case in the other sectors, non-pharmaceutical sectors. That's why it's much more difficult to assess the relevance of in silico modeling for humans in the non-pharmaceutical sectors. The issues, other issues that also have been identified as problems for the PBPK application in non-pharmaceutical areas, also consider the review process of the model. And the challenges in this part results from lack of expertise, lack of experience in PBPK modeling and application among the agencies. So that's why there is maybe not lack, but shortages of the individuals who can lead a project, who can review a model which is submitted, and also the issue that has been raised is, which was already mentioned by Matthias, is that there is a lack of consistency between agencies on requirements for the criteria for acceptance for the model. So the review process is not standardized, not even consistent across the different agencies. So another issue is the number of platforms that are used to build PBPK models for chemicals, and a lack of transferability between the models, between those platforms, which make the assessment of the models even more complex. So to summarize, the PBPK modeling is advanced, so it can be used for predicting, for modeling the chemical behaviors in the body, and there is a willingness across the regulators to accept those new techniques and the results of PBPK modeling. But what is needed is the more confidence in their predictions, and for that, the non-chemical sectors need to develop a quality assessment strategy. It needs to be strict, it needs to be robust, and it needs to be based on the scientific merits, but still be doable. And this, for this purpose, of course, is the integrated input and collaborative input from both sides, which is the regulators and PBPK users or modelers, is strongly required. And this is what I already heard from your presentation, that is already here, and you are working on the quality assessment strategy. And the last statement is that the pharmaceutical sector, pharmaceutical industry can serve as an example of what needs to be done and how it can be done to provide more confidence to the PBPK models to be relevant, to assess the chemicals and the regulatory decisions. Thank you for your attention.